Welcome back to this mini lecture. Mini lecture 10C is going to deal with the special senses known as taste and smell. Taste requires the dissolving of substances and their binding to sensory cells, which are called separate cells. Now, when you read textbooks, these separate cells, these sensory neurons, are given the name gustatory cells because the physiological term for taste is called gustation. We're going to see that our gustatory cells are classified as chemoreceptors. They are capable of binding chemicals which are dissolved in a fluid called saliva. Gustatory cells are specifically located in structures known as taste buds. We have about 10,000 taste buds found on the surface of our tongue, the soft palate, and even down as far as our larynx. Although I would say if you're tasting something by taste buds in your larynx, you have a big problem. You're choking. Our taste buds specifically are found associated with surface projections that are called papillae. On the tongue, there are numerous types of papillae. There are the fungiforms, the circumvallates, the foliates, and we also have another type of papilla called filiform, but these are not for taste. These don't bear taste buds, in other words. They allow us to detect the texture of our food. And the texture of our food sometimes can be just as important, if not more important, than the taste of it. Our taste buds are located on the sides of the papilla. They will be constantly bathed in saliva, and the saliva is there to dissolve our taste chemicals. Now, the taste chemicals are referred to as tastins. We have numerous types of gustatory cells to detect these tastins. The 5 general categories of gustatory cells are going to be for salty, sweet, bitter, sour, and umami tastins. Now, most of you are familiar with salty, sweet, bitter, and sour. You might not be familiar with umami, but this is more of a meaty or savory taste. The general idea is that the tastins are going to bind receptors that are found on the gustatory cells, and they are going to stimulate these gustatory cells to release neurotransmitters. These neurotransmitters are then going to stimulate the remaining neurons in the taste pathway. Here's the thing though, tastins are not specific to the taste bud, they're specific to the gustatory cell. So on a gustatory cell, there has to be a receptor specific for a salt tastin. That gustatory cell is not going to be capable of binding a sweet tastin or a sour tastin unless there is a receptor for it on it. So there's quite a bit of specificity associated with taste. A taste bud is an oval or an onion-shaped body consisting of about 50 neuroepithelial cells, which are referred to as gustatory cells or taste cells. And these taste cells are surrounded by glial-like cells that support them. In this picture of a taste bud right here, we've got our taste or gustatory cells. Here's one right here. I'm going to diagram it in red. That's a supporting cell I'm diagramming right here in red. So these are our supporting cells. Function as glial cells, and their job is to support the gustatory or taste cell right here that I'm going to highlight in green. Here's another one in green, and finally there's another one over here in green. So there are about 50 of these neuroepithelial cells called gustatory cells, and in between those we've got ourselves these supporting cells. You'll see at the apical end of the taste bud we have these structures called gustatory hairs. These gustatory hairs are actually microvilli. They project off the apical end of our gustatory cells, and they project upwards and into the oral cavity through a structure called a taste pore. Now, if it helps you to think of these gustatory hairs as acting of kind of a dendrite, that's fine. They are not dendrites, though. They are microvilli, but they do the same sort of thing that a dendrite does. They receive incoming information. Each gustatory hair is going to possess numerous sensory receptors that bind tastins. So each hair possesses numerous chemoreceptors that bind the chemicals of our food, that bind what are called tastins. Another thing I want you to pay attention to is this structure here that I'm going to highlight in red. Again, I'm going to circle it right next to here. It is called a basal cell. I want you to think of these as being the stem cell of the taste bud. Their job is to regenerate brand new gustatory cells every 10 to 14 days. These gustatory cells are going to synapse with the first order neurons. First order neurons are shown in blue at the bottom of the diagram. And these first order neurons are going to connect to our gustatory cells like this, and then their axons are going to run out towards the central nervous system. And these axons are clustered together to form what we call the cranial nerves. And these cranial nerves, I believe it's 9, 11, and 12, are the 3 cranial nerves that supply the taste buds in our oral cavity. Oh, I was so close. The cranial nerves are 7, 9, and 10. I wasn't anywhere near close to it, was I? A lot of times in anatomy classes, they'll have these little models, and we don't have this one in our anatomy room, but I did kind of like it, so I took a picture of it and I labeled it up for you. It shows you right here the gustatory neuron, shown here in blue, so I'm going to highlight that in blue, and you can even see it forms these little gustatory hairs that project out into the oral cavity. We've got our supportive cells right here that are sort of these, I don't know, pink colored ones. You'll notice that there are these yellow structures here. These are going to be our first order neurons, which are going to be synapsing with the gustatory neurons. And you can see here the axons clustering together. These are going to come out and form the cranial nerves, either 7, 9, or 10. When I first began teaching physiology over 20 years ago, it was thought that there were separate taste buds for sweet, salty, sour, etc., etc. But we now know that's not true. Physiologists now know that each taste bud is a mixture of distinct separate cells that can detect these different kinds of tastins. So a taste bud now is known to be a mixture of neuroepithelial cells, or what are called separate cells, that can detect a wide variety of tastes. Taste buds are thought to contain 3 basic kinds of cells: type 1, type 2, and type 3. Type 1 cells are shown on the periphery of this taste bud that I have on this slide. They are generally known as support cells or supportive cells. They are thought to primarily fulfill the role of a glial cell, in that they support the remaining types of cells within the taste buds so that those types can generate their graded potentials. But research has shown that these type 1 support cells can also detect low levels of salt, specifically low levels of sodium. And in doing so, they can bind sodium, they can depolarize and generate a graded potential, and they can release ATP, which functions as a neurotransmitter, binding the downstream neuron that makes up the taste pathway. In this picture, you can also see type 2 taste cells. These are the ones shown here. They're kind of purple and blue, so I'll circle them right now. These are the ones that are going to bind tastins for sweet, umami, and bitter tastes. Just like the type 1 support cells, they're going to bind these tastins, they're going to depolarize and generate a graded potential, and they're going to release ATP, and this ATP is going to function as a neurotransmitter, binding the downstream neuron in the taste pathway. The type 3 ones, the type 3 cells are shown here. These are these little pink ones there, right? I'm going to circle in green. These are for sour tastins. These are probably the most conventional neuron-like cells we have in a taste bud. They are going to respond to sour tastins. They are going to depolarize. I believe these ones actually do generate an action potential, and they release the neurotransmitter serotonin, which then goes and binds the downstream neuron in the taste pathway. Before we leave, sorry, I'm going to erase this here. I'm going to erase this red one, and I'm going to draw in, because I forgot to include the green taste bud here. All 3 of these are your type 2 taste cells. So sorry about that. The physiology of taste can be a little complex, but it does rely on the binding of a ligand, which is the tastant, binding a receptor, a sensory receptor, located on a gustatory cell's hair. Now, for the most part, most gustatory cells, the tastant binding is going to lead to a change of receptor potential. It's going to lead to depolarization of the gustatory cell, and it's going to produce something called a graded potential. In the majority of gustatory cells, the tastant produces a sub-threshold graded potential. You don't get an action potential, but that graded potential is still enough to cause the release of, say, ATP or a neurotransmitter like serotonin. Where the confusion comes is that the mechanism of how this graded potential develops differs based on the tastant. It differs on whether you're looking at a type 1, 2, or a type 3 gustatory cell. So for example, the type 1 and the type 3 gustatory cells, they'll bind ion channels, and those ion channels will open, and then the sodium and protons will enter into the type 1 and type 3 gustatory cells. For the type 2s, this is a really different mechanism. The tastants are going to bind a G protein coupled receptor, turning it on, and you're going to get a series of events within the cell that is eventually going to produce a second messenger inside those type 2 taste bud cells, and that second messenger is going to open up an ion channel. Then you're going to get depolarization. So for the type 1 and type 3, your graded potential is produced more of a direct way where ions flow in and cause depolarization. For the type 2 taste cells, it's a little bit more indirect. You're going to get a G protein coupled receptor opened up, and then eventually ion channels will open up and ions will flow in, and you'll get a graded potential. No matter how you get there, that graded potential is going to cause the release of something from those taste cells. From the type 3 taste cell, you're going to get the release of serotonin. From the type 1 and type 2 taste cells, you're going to get ATP being released, which is going to function like a neurotransmitter. And these things like serotonin or ATP are going to bind to and stimulate the first order neuron that attaches to the taste bud. That is now going to generate an action potential. And you're going to get that action potential traveling along the first order neurons, which form cranial nerves 7, 9, and 10. Taste buds do adapt relatively fast. You can get complete adaptation anywhere from 1 to 5 minutes. This explains why something which has a very bold taste when we first taste it kind of dims over time. And our thresholds for tastes do vary among the 5 primary tastes. We are most sensitive to bitter, which is why we put these bitter chemicals in poisons. We are least sensitive to salty and sweet chemicals. So let's go over it again, because the mechanism for that taste cell depends on the taste cell and the tastant. Let's start off with the type 1 cells, which are going to respond to salty foods, specifically low salt content. The sodium ions are going to bind and open up a sodium channel. The sodium ions are going to enter. You're going to get depolarization. You're going to get a graded potential, and you're going to get the release of ATP, which is going to function like a neurotransmitter. This is more of a direct method. You're going to get a very direct method for the type 3 taste cells. You're going to have protons, which are going to bind these channels, open up these channels, enter into the type 3 taste cell. You're going to either get a graded potential, but some type 3 cells have seen to depolarize to threshold and get an action potential. Whether or not they actually hit an action potential, these type 3 cells resemble neurons in that they actually do release a bona fide neurotransmitter. It's usually serotonin. And they do so by little vesicles that fuse with the plasma membrane and then secrete those neurotransmitters out. That serotonin is going to bind the first order neuron and trigger an action potential. So out of all these gustatory cells, the type 3 cells act more like a neuron. Where the confusion comes in again is the tastants for sweet, bitter, and umami. These are going to bind the type 2 cells. One is specific for sweet, one is specific for bitter, and one is specific for umami. They do not cause ion influx directly. Instead, these tastants bind G protein coupled receptors found on those gustatory hairs. You're eventually going to get ion influx and depolarization. This is because the G protein coupled receptor, like I said in the last slide, is going to set up a series of events. So the tastant's going to bind the receptor. The receptor is going to activate its partner, which is called a G protein. The G protein is going to activate its partner, which is called an effector. The effector is going to produce a second messenger like cyclic AMP, and it is the second messenger that opens a ligand-gated ion channel, causing ion influx, and a graded receptor depolarization is going to result. So this is a much more indirect system, is it? But eventually you do get ion influx, you do get depolarization, you do get a graded potential, and type 2 cells release ATP. This G protein signaling is something that we saw way back at the start of the course when we talked about cellular communication. So I think I may have used a similar figure like this, but let's go over it again because it's not really something you're familiar with as a physiology student. Here's our ligand. This would be a tastant. This would be your sweet, salty, or your sweet, bitter, or umami tastants. It's going to bind the G protein coupled receptor. This is going to turn on the G protein coupled receptor, and that's going to activate the nearby G protein. See how they physically interact with one another? This is going to turn on the G protein. The G protein is then going to physically bind and activate the effector. So here they are down here, physically binding, and now the effector gets turned on. And now this effector is going to lead to the production of your second messenger. This second messenger is then going to bind an ion channel, and ions are going to flow in, and you're going to depolarize and generate a graded receptor potential and release ATP. Let's take a look at the gustatory pathway, or what is known as the taste pathway that connects with these taste buds. The gustatory cells synapse with what are called gustatory fibers. These gustatory fibers are considered to be first-order neurons. The axons from these first-order neurons are going to form very specific cranial nerves. They're going to form the cranial nerve 7, 9, and 10. Cranial nerve 7, which is the facial nerve, is going to serve the taste buds found on the anterior 2/3 of the tongue, so the front 2/3 of the tongue. Cranial nerve 9, or the glossopharyngeal nerve, is going to serve the posterior third of the tongue. And cranial nerve 10, which is the vagus nerve, serves taste buds found in the palate and even as far down as the epiglottis. These first-order neurons are going to come in the brain stem, and they are going to stop at the medulla oblongata. Some tracts can run directly to areas of the limbic system at this point. These tracts that run to parts of the limbic system are thought to be responsible for taste aversion, which is the conscious and strong connection between taste and an emotion. There are very strong connections between our limbic system and this taste pathway. This explains why you might absolutely hate something like the taste of raw tomatoes, which is me. That is a taste aversion that I possess. You may possess other ones. So our first order neurons come in and stop in the medulla oblongata. They make connections to the limbic system. But in the medulla oblongata, these first order neurons are going to connect to their second order neurons. The second order neurons are going to run from the medulla oblongata and stop in the thalamus. It's at this point they're going to synapse with their third order neurons. And the third order neurons are going to exit from the thalamus and run to the primary gustatory area, which is located in the parietal lobe of our cerebral cortex. Processing in this primary gustatory area is going to provide conscious perception of taste. You're not going to know what you're tasting, you're just going to know that you are tasting something. That information is then going to get sent out to surrounding association areas in the cerebral cortex, and only then will we be able to identify that what we are tasting is sweet or salty or bitter or something like that. In many ways, smell is similar to taste because smell is a chemical sense. You are going to have the dissolving of chemicals in a fluid. Those fluids are going to bind separate cells, and you are going to eventually result in the generation of an olfactory signal and processing somewhere in the central nervous system in the brain. But there are some differences. So let's start off with the basics. The sensory neurons for smell are separate cells. They're referred to as olfactory cells. This is because the physiological term for smell is olfaction. These olfactory cells are anatomically located in what is called the olfactory epithelium. This is found in the nasal cavity just above the superior nasal concha. So there it is right here. Each olfactory cell is going to project multiple cilia down into the nasal cavity. As a matter of fact, I read these are the only neurons in the body that are directly exposed to the external environment. So how about that? In this picture down here, you can see these cilia right here. There they are being projected down. Let me do this in blue here. So projected down into the nasal cavity. I want you to think of these projections, these cilia, as kind of acting like dendrites. They will have receptors on them for the binding of the chemicals of smell, what we call odorants. So odorants are going to bind receptors on the cilia, This is going to affect the activity of these olfactory cells. We're going to see how in a second. But these olfactory cells are going to run up through the cribriform plate of the ethmoid bone and will synapse with the cranial nerve 1. And that's what's shown here. You can see these olfactory axons running up through the cribriform plate, and they are synapsing with the cell bodies of the neurons that make up olfactory nerve, cranial nerve 1. You know, in a way, the olfactory epithelium to me reminds me of a taste bud that you've just kind of unfolded. You've got your supportive cells. These are columnar epithelial cells that function sort of like glial cells. Their job is to support the olfactory cells so that they can do their job. You've got basal cells, which are stem cells. Their job is to replace our olfactory cells on a regular basis. But the difference is between a taste bud and an olfactory epithelium is that in a taste bud, the actual cells binding the chemicals and responding and depolarizing, these are neuroepithelial cells we call separate cells. In the olfactory epithelium, the olfactory cells are actual bona fide bipolar neurons. They are neurons. They generate action potentials. They release neurotransmitters. Here is one of those. I'm going to try and highlight it here in red. Here's one. This is its cell body. Because it's bipolar, it has a long dendrite which projects down, and at the end of this dendrite, there are little tiny cilia which project into the nasal cavity. And then at the opposite end, you have the big long axon which comes up and is going to synapse with the neurons that make up the olfactory bulb. The cilia are going to possess chemoreceptors that bind the odorants. These chemoreceptors are really a lot like the chemoreceptors we find in taste buds. They are sensory receptors. Their job is to bind these odorant chemicals and then result in depolarization. If you want to think like in taste, these cilia sort of act like dendrites to receive incoming sensory information, that's fine, but they are not. In taste, they are microvilli, and in smell, they are cilia. Because again, these are bona fide neurons. They are called bipolar neurons. These olfactory cells are the first order neurons in the olfactory pathway. In taste, the separate cells synapsed with the first order neurons. In smell, the first order neurons are the actual olfactory cells. We do have structures which are called olfactory glands. That's this thing right here. I'm going to try and circle it right there for you. These olfactory glands, their job is to produce mucus because that is the fluid that our odorants are going to dissolve in. Both our olfactory epithelium and our mucus glands are innervated by cranial nerve 7. The mechanism for olfaction reminds me of the same mechanism we saw with the type 2 taste cells. It's more of an indirect system. So you've got odorants shown here, down here as these little red dots that are dissolving themselves in the mucus. They're going to bind their chemoreceptors on these cilia. And these chemoreceptors are G protein coupled receptors. So they're not ion channels. They are G protein coupled receptors. That is then going to activate the G protein. The G protein is going to activate the effector, and the effector is going to produce the second messenger called cyclic AMP. Cyclic AMP is then going to open up ion channels, and you are going to have ions flowing into the olfactory cell, and you get depolarization, just like in taste. But unlike taste, you are actually going to get an action potential. Remember, in taste, the majority are type 1 and type 2 cells, and a lot of the times our type 3 cells just go up on a graded potential. In olfaction, these are neurons. They are going to depolarize the threshold. They are going to generate an action potential, and they are going to release a neurotransmitter. The neurotransmitter, I believe, is glutamate, and that neurotransmitter being released right here is going to then bind the second order neurons who actually form the olfactory bulb. And then that impulse is going to be sent down their axons. Axons of these second order neurons is going to form cranial nerve 1, which is the olfactory nerve. And then beyond that, don't worry about what the third order is. Beyond that, the olfactory pathway gets really weird. The olfactory pathway is the one sense pathway that does not relay through the thalamus. Your other senses, like your general senses and your other special senses, will relay themselves through the thalamus. This is not the case in olfaction. There are a couple other fine distinctions between the olfactory pathway and a typical sense pathway that we learned in the previous lecture. For example, the first order neuron is the actual olfactory cell in the olfactory epithelium. When we looked at taste, the first order neuron synapsed with the gustatory cell. In olfaction, the first order neuron is the olfactory cell. The second order neurons are going to form the olfactory tract. Each tract is going to travel to multiple areas of the brain. However, most second order neurons are going to end at the primary olfactory cortex located in the temporal lobe. This is where conscious awareness of smell begins. Third order neurons are then going to take that information and bring it to other areas of the cerebral cortex so we can begin to identify what that smell is. And the third order neurons are also going to allow us to make extensive connections between the olfactory pathway and the limbic system. Out of all of our senses, smell is the fastest adapting sense. You will get 50% adaptation in 1 second. You can get complete adaptation in a minute. This means the intensity of what you are perceiving will go down in second 2 by 50%. By the time 60 seconds rolls around, you may not smell it at all. And you may experience this a lot of times when you go into a room and you say, "Ooh, what's that smell?" And somebody looks at you and says, "I don't smell anything." There is a very low threshold to stimulate olfactory neurons. Only a few molecules or a few odorants need to be present. We are especially sensitive to like rotten egg smells that are produced from methyl mercaptan. This is why we add that chemical to natural gas as a warning, because natural gas has no smell and we're really good at detecting rotten smells. By the way, there is a condition called hyperosmia in which people have a keener sense of smell than others. The opposite to this is called hypoosmia, which is a reduction in the sense of smell. Hyperosmia actually can be seen in women around their time of ovulation. This is an evolutionary holdover of females being able to smell their mate so that they can copulate and have babies. Now, we are done with smell, but I want to give you one more interesting fact. There was a study that was done with women who were ovulating. They got their mates to wear a t-shirt for something like 2 weeks without washing it. And they put that t-shirt in and amongst, 13 or 14 other stinky t-shirts from 13 or 14 other stinky men. And pretty much without fail, the ovulating woman would identify the t-shirt she liked the best as being the one belonging to her mate. Let's move on to the next mini lecture in this series, where we're going to talk about hearing and balance.